So let's take a look at carbohydrates and the types of molecules that they form. When we're looking at carbohydrates, you're looking at three elements. You're looking at any molecule that has basically just three elements, C, H, O. There are exceptions to every one of these rules, but you have carbohydrates are going to have generally a 1 to 2 to 1 ratio. So you're looking at what I like to think of as CHO, right? So carbohydrates have only CHO in them. Now, something you might want to think about is that when you're talking about carbon as a carbon bond, and an oxygen, a double bonded oxygen. The double bonded oxygen could be within a carbon chain, right, like you see here. Or it could be, and an example would be something like an acetone, right? And you know it as nail polish remover. And it has 3 hydrogens. I'm not putting the H's. It's connected to a center carbon that has double oxygen and another 4 hydrogens. So this specific type of bond here, C double bond O, in the middle of a chain, is known as a ketone. Now, ketones, it's moderately important that you understand what a ketone is, as opposed to an aldehyde. An aldehyde is going to be this, it could have the same exact structure, same number of hydrogens, except that the double bond could be at the end. And this would be more of an aldehyde. Now, these differences, they seem small when they're taken out of context, but you have to understand that when you're dealing with chemical reactions, biochemical reactions, the position of these functional groups can make the difference between an enzyme working or not working, or a drug working or not working. So this C double bond O, they're both C double bond O, these groups, but it's just where it is, whether it's inside the structure or it's at the end of the chain. So why is that important when we're talking about something like carbohydrates? Well, it'll come into focus in just a minute. There are a number of carbohydrates. We can start with the first, and we'll talk first about the monomers. Remembering that monomers are the single units that, when put together, make polymers. And we can take a look at something like glyceraldehyde. Now, glyceraldehyde, I think, is going to be one of those important molecules. If you get to see the structure, you understand the structure, I think you'll end up appreciating some of the other structures that are based on glyceraldehyde. So get to know this 3-carbon sugar. It's a very important 3-carbon sugar. In photosynthesis, you'll see it again. Certainly, it's important in triglycerides, and it's important in phospholipids. So here's this 3-carbon sugar. It has this aldehyde. It's an aldehyde. Why? Because the functional group is at the end, not in the center. And then you have an OH, an OH. So again, the name of this is glyceraldehyde. And it's when you break down glucose in your cells, the very first thing that you make is glyceraldehyde. So this is a very key component in a lot of different biochemical pathways. So getting to know glyceraldehyde is a good investment of your time. 3 carbons, it's an aldehyde, so it has a double bond O at the very end. The next that you'll look at is ribose. Now, I'm going to draw ribose as all of these. I'm going to draw them as as linear molecules, they can also form rings. Not the glyceraldehyde, but the ribose, the glucose, and the fructose can all form rings. And we'll look at those rings in a little bit. But the bottom line is ribose is another sugar. Now this is a 3 carbon sugar. 1, 2, and 3. But we can have a 5 carbon sugar, like ribose. 2, 3, 4, and 5. And this 5 carbon sugar is also an aldehyde. Now notice that this is, as we discussed with the general formula, for every 1 carbon you expect to see 1 oxygen. So when we're drawing these out, we're thinking, or at least I'm always thinking, well, that's it makes sense to me that this is a sugar, since I see that for every carbon I have an oxygen and I have double the number of hydrogens. So this is ribose, also an aldehyde, right? It's found in RNA, right? It's a ribonucleic acid. It's found, so this is the ribose. If you take off one of these oxygens, it would be deoxyribose. But this is the sugar that RNA is based on. One of the main components of the nucleotide RNA, an RNA nucleotide. Again, 5 carbons. 1, 2, 3, 4, and 5. That's not really how we number them. I should number them the way we're supposed to number them. We need to number them from the oxygen, from the double bond, from that aldehyde group. From wherever that double bonded oxygen is, we're going to number them. So let's go ahead and number them. And really, numbering doesn't really become that big a deal until you form rings. And by numbering them, it's going to allow us to discuss where certain things are. For instance, in deoxyribose and ribose, there are specific carbons where specific connections are made, and something that you're going to need to identify, so being able to number them is useful. This is ribose. Now, glucose is a more familiar molecule to you, I think. It's also an aldehyde, and it is 6 carbons. So here you have 1, 2, 3, 4, 5, 6. And again, being a sugar, it has 1 oxygen for every carbon, except it's an aldehyde, so there's a double bonded oxygen up here, and there's a hydrogen and down this way. These are all hydrogens. Double the number of hydrogens for every carbon. Again, a good rule of thumb. This is glucose. So 1, 2, 3, 4, 5, 6 carbon sugar called glucose. Metabolically, it's an incredibly important molecule. It is glycolysis is the first step in cellular respiration. And all right, so there's glucose. Now, fructose, which is actually much more difficult for us to digest, and it turns out that fructose is not a sugar that we can easily process. We can process it, but in large amounts it can cause metabolic issues. It's also a 6 carbon sugar. It's one of those isomers. Glucose and fructose are isomers. They're structural isomers. And the interesting thing, they're both hexoses, so both of these sugars. Both of these sugar monomers are indeed 6 carbons. The difference is that that fructose is not an aldehyde. It's a ketone, so it has a double bonded oxygen here. Now, how does that change it? Well, it changes how you can process it. It's much more difficult for us to process fructose in our bodies than it is glucose. It also makes a difference in our taste, obviously. I think you would agree that fructose tends to be a little sweeter. It gives fruits their tastes. So when you're looking at fructose versus glucose, the difference is so small. It's almost hard to understand how it could make such a big difference. Is this double bond O being on the second carbon here instead of the first up there, right? So just changing it from an aldehyde to a ketone changes how we can process these two sugars. So fructose is that much different from glucose simply because the double bond is not on the end of the molecule, but rather in the center of the molecule. So it is a strange thing that something so small can make such a big difference. And in fact, it could be eating too much fructose could lead to pancreatitis and problems in digestion. Obviously, too much glucose can lead to diabetes, but too much anything could harm you. But fructose has we have a lower tolerance for fructose than we do for glucose. All right. Well, when we talk about making a ring, the ring structure is made by. Let me see. How can I? There it is. The ring. You go from linear to a ring structure simply by shifting some of the bonds around. And actually these molecules form rings at equilibrium. So they actually go from ring to straight chain back to ring again all the time. And depending on the conditions, you'll find them as rings or as linear molecules. And the only difference is that one of the hydroxide groups, the one depending on which molecule you're dealing with, in the case of glucose, it would be taking the second carbon, the hydroxide group here. I should just make a little asterisk. This second carbon hydroxyl group will actually go and form part of the ring. It'll actually close up. When it closes up, it makes a bond with that first carbon. It'll actually attack that first carbon. So let's go ahead and do that. Let's see. You'll see that this. I'm going to show you here on a micro scale what's happening. Just focusing on those bonds. Normally it would have a single bond. There'd be a double bond O, and there'd be an H coming off here. And then over here you have the hydroxyl group. That's an OH group, right? From the second carbon. Second to last carbon, rather. I put it on the wrong one here. So the hydroxyl group from the second to last carbon, which would be here, the fifth carbon, that hydroxyl group is going to come and, like a snake eating its own tail, is going to make a bond then with this carbon. Now, in order for it to make that bond, carbon can only make 4 bonds. This would make it 5. Well, it can't do that. So, and oxygen can only make two bonds, and right now it has a bond to the to the carbon that's over here. So it needs to in order for this to make a ring, this oxygen has to stay, right? So connected to this carbon as it connects to this carbon. In order to do that, this hydrogen has to leave. So the hydrogen leaves. Now when that happens, you'll see here that this has a double bond to this oxygen. And carbon can't have 5 bonds. It can only have 4. So it's going to lose one of those bonds. It's going to lose one of those bonds. So now it has 4 bonds. And but now the oxygen needs another electron, or it doesn't have a full octet. Where it used to have a full octet because it was double bonded to the carbon, now it's not. So, but there's this hydrogen that was they got kicked out of here, right? There was a hydrogen that got kicked out of there. Well, a hydrogen comes in here, and what you form is an OH group, and that's how you form the ring. Of course, the rest of the sugar is still there. So let's go ahead and draw the rest of that sugar. Let me erase the hydrogens because it left over here this is the 5th carbon sugar. It left that. All right, the first carbon now is part of the ring. That is no longer an aldehyde. It's carbon here. There's an OH group. Remembering that each of these has an OH. Another carbon. So that's the 2nd carbon, that's the 3rd carbon. There's an H and OH. And they tend to alternate, like one OH going down, one up. Then you have, and that has to do with the repulsion of the electrons and the molecular geometry of chemical bonds. We're not going to get into that. But there's your 3rd carbon. Your 4th carbon then, of course, is the 4th carbon has an OH here again, and a hydrogen sticking out. That's going to be connected to your 5th carbon, which is already part of the ring, as you see here. So that's what helped close it off. Then sticking up is the 6th carbon that has 2 hydrogens and a hydroxyl group. So every carbon has, I have to go ahead and draw the hydrogen here. Every carbon has 4 bonds, and you can check your work by looking to see every carbon has 4 bonds in it. This bond is not really drawn well enough. It's pretty short, but if you drew it, if I had drawn it correctly, you'd see there was an oxygen. It's connected to the first carbon. You have an oxygen connected to the first carbon, connected to the 2nd carbon, connected to the 3rd carbon, connected to the 4th carbon, connected to the 5th carbon. It's a little short on that end. I need to get a better tip. So there's my 1st carbon, 2nd carbon, 3rd carbon, 4th carbon, 5 carbon. And the reason it closed up is, again, it's that movement, the loss of the aldehyde, right? The double bonded aldehyde group. You lost that. And that bond then formed part of the ring. Being a ring molecule, it gives it different properties, and sugars tend to form these rings. And here's this 6 carbon sticking out of here, and again that has an OH group and 2 H's. So we're taking a look at these monomers. Now, when I drew these, I want to remind you that I have left off the HOH, and that those always these sugars have these OHs, hydroxyl groups, right, that stick out, and the hydrogens are there. Often we when you see as we move along, we'll kind of drop all these details, but you're expected to know, and then the exam and the various AP exams, they're going to expect that you understand when you look at any drawing of glucose that you have all these carbons, all these hydrogens, all these oxygens are all present in these molecules, these carbohydrates. And you can count them, but there would be, the ratio would be CHO. So that's a ring structure. It would see to one carbon to two oxygens to one one carbon to two hydrogens to one oxygen. So those would be ring structures. And when you're dealing with the ring structures, we can put their monomers, and so a monomer of sugar would look, something like this with the oxygen there and oxygen there, and you have a hexagon, and often the hexagons will be drawn like so. And you're going to have a hydroxyl group sitting here. Now, anytime you have a hydroxyl group, there's an opportunity. And you'll see when we draw the other, let's go ahead and draw fructose. Now, fructose, when it makes a ring, it's 6 carbons as well, but as a ketone, it doesn't form a 6-carbon ring, right? And if it were an aldehyde, it would form a 6-carbon ring, very much like you see here with glucose. But because it's a ketone and not an aldehyde, and that double bond being on the second carbon instead of being at the end carbon, what you end up forming is a 5-ring structure. So even though it has 6 carbons, it's going to form a pentose. It's going to have 5 sides. Each corner, remember, is a carbon. And here on this side would have something like so, and you'd have OH, and then you'd have an OH up here or something like that, and there'd be a hydrogen down here, and there'd be an OH here, and an H up here. Remembering, again, it's a sugar, so on every carbon, you're going to have this OH and H. Just go back and look at all the various sugars, the trioses with 3 carbons, the pentoses with 5 carbons, the hexoses with 6. No matter what, you have carbon, oxygen, and hydrogen. Again, I'll write it up here, CHO, right? Carbon, hydrogen, and oxygen. And you're just going to look for a 1 to 2 to 1 ratio. That's a general formula. So the key here is that you're going to have this OH group sitting here and another OH group sitting there. There's an opportunity there that life has been able to take advantage of. An opportunity to link 2 large structures together. And let's take a look at what's going to happen here. I'm gonna go ahead and let you think for a second before I answer, or before I tell you. If you have an O here and an H, and then you have an H here, what could possibly happen that would allow you to bond this oxygen, right, this oxygen, or this carbon to this oxygen? Could you use this? How can you bond this carbon to this oxygen? Remember that this oxygen only has two bonds. One of those bonds is to this carbon. Also remembering that when you're looking at these, the corners are carbon. So 1, 2, 3, 4. I missed one there. Sorry to say. It's fructose, so that always does that to me. 1, 2, 3, 4, 5, and 6, right? 6 carbons. So what are you going to do? How are you going to connect? If this oxygen can only make 2 bonds, or this carbon can only make 4, it has 1, 2, 3, 4. This oxygen can only make 2, and one of them is to this fifth carbon, and one of them is to the hydrogen. How can you connect this carbon to this oxygen? Well, this oxygen would have to lose one of the bonds. If it lost the bond to this carbon, then it would just fall off, and you wouldn't connect these two. You wouldn't be able to connect glucose to fructose. If you broke this hydrogen off, then the oxygen would be connected to this, but there's still no way to connect it to this ring. So what's interesting is if you take these two out, if those two come out, you have OH and H plus sitting out here. OH negative and H plus sitting out here. As they leave oxygen and they leave, there's no bond there now. Now you're going to let this oxygen, the ability, you leave this oxygen, the ability to form a bond to this carbon. This carbon to oxygen to carbon. Carbon to oxygen to carbon. Where the OH and H come out and they form, out comes water. So what is it called when we take water out? I hope you know what the answer is there. When you take water out of something, there's 2 words that you've learned in middle school probably, and you certainly heard in high school biology. When you see water coming out, you would think of 2 words, at least I do: dehydration, water leaving, coming out, dehydration. And also I would think condensation. Those are 2 words I would be thinking. Condensation, dehydration. So if you're thinking about those 2 words, condensation, dehydration, water's coming out, what are you doing? You're making something. You're making something new. You had glucose. I'm just going to put GLU. And you had fructose and put FRU. And you're going to connect them to form something new. We call this then a synthesis. And so it's a dehydration synthesis. Some people might call it a condensation reaction. But it's a dehydration synthesis. And what you make is a sugar, glucose. Again, I'm just going to draw this without all the rest of all the extra, just for the sake of simplicity. Connected to an oxygen that then is connected to a fructose. You know what? I'm not going to put that extra stuff there either. Just leave the H. I'll leave the carbons because I have to put these because these are part of the carbon backbone here of the 6-carbon sugar. So it's not ribose. Ribose is only 5 carbons. This is actually 6 carbons. I suppose if I was going to do that, I should put the extra carbon here as well, right? Because this is still oxygen. So when we're looking at this structure, this is called a disaccharide. Right? It's a disaccharide. It's a disaccharide known as sucrose. This is table sugar. This is what you eat all the time, the white sugar that's in the bag. So disaccharides are found in nature a lot. There's a number of them. There's another disaccharide I'm sure you've heard of. I'm going to go ahead and say that there's a disaccharide called lactose that I know you've heard of. And that's a sugar known as a 6-carbon sugar known as glucose connected to a 6-carbon sugar known as, you know what, let me do it the same way, connected to a 6-carbon sugar known as galactose. I wrote out galactose because I think it's one you may not remember how to or have seen. So here, sucrose is one where you have fructose, a 6-carbon sugar, and a glucose, a 6-carbon sugar. The difference is the ring here is a 5-sided ring, where galactose is actually a 6-sided ring and glucose is a 6-sided ring. And they form this, by the way, we call this a glycosidic bond. The bond that holds the sugars together. And so this sucrose and lactose, these two examples of a disaccharide, are important. In fact, when you look at lactose, there are people that are lactose intolerant. They can't break this particular bond between glucose and galactose. These people can eat sucrose, but they can't eat lactose because of an enzyme that breaks one down, but they can't break the other down. In fact, the enzyme that breaks lactose down is known as lactase, is found mainly in babies. And since milk is made for babies and it was never actually meant to be used as a food source for adults. So when we look at glucose and sucrose, or lactose and sucrose, when we look at dehydration synthesis, this process of dehydration synthesis is going to be repeated over and over again. When you build 2 biomolecules, you're always doing dehydration synthesis. You're taking advantage of the OH groups. You're taking of the hydroxyl groups. You're taking a hydroxyl group and a hydrogen. You're putting them together to make water, and you're using the missing bonds to connect 2 parts, 2 units together. And the units can be different. For instance, between glucose and fructose, they are both all of these are connected through glycosidic bonds. But glucose and fructose are fundamentally different in the sense that this is a ketone, and this is an aldehyde. So this has a double bond oxygen on one end, and the double bond oxygen is in the center of the ring, or closer to the center, the second bond. These are isomers, structural isomers of each other, and because of that, their shape is different. And what your body's able to do with it is different. And so when we look at sucrose, is that glucose and fructose put together. But when you look at lactose, is two different units put together. You have glucose and galactose. They form a different disaccharide, but they're both disaccharides. All right, so disaccharides are great, but polysaccharides are actually much more interesting. Again, we can connect them together. And it's these subtle geometric differences in three-dimensional space that can make the difference between being able to break down something and digest it or being sick. There's a disease called Galactosemia. And galactosemia is an inability to break down this sugar, galactose. So the babies are able to drink milk and break down the lactose into glucose and galactose, but then they can't digest the galactose, even though it's just a 6-carbon sugar, which is just different simply by the type of connections that it has to the carbons and the oxygens. The baby can't break down the galactose, and that galactose builds up in the blood and causes all kinds of problems. In the past, it would mean disease and death. Today, we know there's a treatment with simply managing the diet to avoid galactose. It helps a great deal, but these babies can't digest galactose. So it's important to understand that these differences, even though they might seem subtle and might seem almost silly and really something that's not important enough to memorize or to learn, it actually turns out to impact people's lives. So when we look at something like starch, starch is a polymer. It's a polymer now. Now, it's not a dimer. Polymers are many, many units connected to each other. And when you have a polymer of sugars, specifically glucose, when you have glucose polymers, there's several different forms of glucose polymers several different ways you can connect these glucose molecules together to form different structures that can be used for different reasons. So if you have a bunch of branched, linked glucose molecules, and those glucose molecules are linked in a 1 to 4 orientation, meaning when we look at the carbons, it would be carbon 1, this being the first carbon, carbon 1, connected to, it's not really working out that well, but whatever. For some reason, this oxygen here, what you get is a molecule then that's connected from 1, and this is 1 here, this is 1, carbon 1. But over here, this is carbon 1, this is carbon 2, this is carbon 3, and this is carbon 4. So this is a 1 to 4 linkage. So that 1 to 4 linkage, if it's branched, if it has a lot of branches, and it just keeps going, so all the carbons are linked on that branch, all the carbons are linked on this branch, and then they branch again, and a bunch of carbons are linked here, and a bunch of carbons are linked there. This kind of branching is called amylopectin. They're both starch, but amylopectin is the branched form of starch. Where the non-branched form is called amylose. So amylopectin versus amylose. They're both the form of starch, both connected 1 to 4. Now, when we're looking at cellulose, you still have that 1 to 4, but now instead of having what's called an alpha linkage, so this is alpha, you have a beta linkage. Now the beta linkage is slightly different in that you alternate. Like for instance, in this one, in the alpha, you're going to have a hydroxyl group continuously pointing downward here at the second carbon. So I know this sounds silly. Again, just very subtle differences. But here's an OH and here's an OH. Therefore, all the CH2OHs here are pointing up. Where a different form, the beta linkages, would have these 1 to 4 connections. Would have them alternating. They would alternate. So this here would key up, CH2OH, and this would key down your OH group here, where the next one would key up to OH and down here to CH2OH. So what you have is an alternating, the OH up, the OH down, and an alternating CH2 up and CH2OH down. So believe it or not, that difference makes this stuff up here a food storage. This is for storage of food that we eat all the time. We steal it from plants, but it's a plant food storage. Plant starch is found in plants. It's food storage. I don't know why I put starch. Food storage. Where this form of linkage forms something called cellulose and is used for structure. The plant is going to use it to create its stem, its woody stem, its ability to hold itself up. So the plant is going to use cellulose for structure, and the plant is going to use glucose, or this up here is the starch is, and either amylose or amylopectin is going to use it for food storage. Humans will use this for energy. And humans will use this for structure. We'll tear down trees and we'll make wooden stools and wooden houses out of them. So that's pretty much the idea behind a polymer, this idea of dehydration synthesis, this idea that you have this disaccharides, and the different monosaccharides can influence give each disaccharide a different set of properties that can then specify whether an individual can digest it or not. Plants make starch to store their food. We can eat that starch. Cellulose is also made by plants simply by reversing the sugars, flipping the sugars every other bond, makes cellulose virtually indestructible by most organisms. We can't digest cellulose, neither can the plants. The plants can't break themselves down to make food, even though it's made of sugar, even though it's made of glucose. There are some fungi and bacteria that have enzymes that can break this link, but no animal or plant has this ability that I know of anyways. Yes, termites and termites can break down cellulose, but it's not, and so can cows, but neither cows nor termites actually can break cellulose down on their own. They need bacteria that live in their guts to break down to break this glycosidic bond so that they can take advantage of the sugar. Now, humans can connect sugars like glucose into polymers as well. And humans, though, they don't do a 1,4 linkage. They don't go 1,4 the way you see here with starch. All right, humans use a 1,6 linkage. So if you think about it, that's 1, 2, 3, 4, 5, and then 6 is sticking out. Remember, it's CH2OH. So it goes from 1, the carbon 1, to the 6. It uses a linkage here in this direction. And by taking those two glucose molecules and doing dehydration synthesis and connecting that oxygen and that 6 carbon together to the first carbon, remembering that these here are oxygens as well. I just drew a closed ring as a convention. By convention, we assume that everybody knows that there's an oxygen in the ring. Making that connection and repeating that, we can form something called glycogen. And that's our form of sugar storage, food storage. It's a quick release. It's a little bit shorter term than fat for energy storage, and it's longer. So you can call it short-term storage or medium-term storage of sugar, and glycogen is just one way we can do that. Liver, in humans, we put our glycogen in liver. Other organisms, I think, do too, where a glycogen is simply used by animals, where starch and cellulose are used by plants. And of course, humans eat starch, but we're stealing it from them. It's not made for us. All right, so that's it for carbohydrates. It took about 48 minutes to get through it, a little bit longer than I wanted to, mainly because I'm exhausted. Sorry about that, but I hope I was able to cover the chapter pretty well. We'll cover it in activity in class, and I'd like to make a similar one that's hopefully going to be more to the point and shorter for each of the other biomolecules. All right, bye.